Hello everyone, my name is Kayla Woodson and I am a student in the Forensic Science program here at OSU Center for Health Sciences. Today I will be talking about my research project, which is the study of time-dependent degradation of mRNA encoding the SPRR3 and S100A9 gene markers in saliva stains. Body fluids found at crime scenes can carry mRNA, and this mRNA may be beneficial to use when approximating age of the stain. However, RNA is less stable than DNA, but one common method for mRNA analysis is reverse transcription PCR or RT-PCR, and previous research in this laboratory has shown the relationship between age and RNA degradation in both blood and semen stains. With this project, we wanted to determine whether reverse transcription and real-time qPCR analysis of specific mRNAs in aged buccal swabs can be used to estimate the age of the sample. To do this, buccal swabs were collected and stored in a box to dry in a room temperature environment for specific time intervals, which included time 0, or when the sample was just collected, to 1 week, 2 weeks, and 3 weeks. Then RNA extraction was performed along with inhibition removal to further purify the product, followed by reverse transcription to cDNA. This cDNA was diluted to appropriate amounts to prevent oversaturation during qPCR analysis, and this dilution depended on the total amount of product collected after both extraction and reverse transcription occurred. The following primer sites were used from 5' end to 3' end of the strand. For SPRR3, they were 15, 594, and 768, and for S100A9, they were 27, 310, and 453. Then, real-time qPCR was performed using the QuantStudio 5 system, and this data was analyzed and graphed using Excel. This first graph shows the CT values collected for the SPRR3 marker, and on the x-axis, the dates show when the samples underwent analysis along with their respective dilution amounts, and the y-axis shows the CT values. As you can see, as time from when the sample dries increases, the CT values seem to increase, and this is especially seen with the last 4 samples in the graph. This next graph shows the CT values collected for the S100A9 marker, and here the CT values appear to stay relatively constant even as the sample age increases, which is different than the results seen for the SPRR3 marker. Results so far show that as time between sample acquisition and analysis increases, the CT values for the SPRR3 marker increase. On the other hand, for the S100A9 marker, CT values stay relatively constant. We also looked at the difference between the 5' end and 3' end of the strands, or the delta CQ value, which was not shown here, but this showed no relationship for either marker. Future research will include samples aged up to 4 weeks and will also include analyzing the housekeeping gene ACTB, and since this is still in the experimental phase, further research will include more samples for better data analysis. Here are my references that I used for this presentation, and thank you to both Dr. Jun Fu and Dr. Robert Allen for their help and advice in the laboratory. Thank you so much for letting me share my research with you, and I hope everyone has a great day.